1011100. This is Biostatistics Core Special Topics Talk number 14. What's the deal with machine learning? I'm Mark Williamson for the DaCCoTA Project at the University of North Dakota. So the goal of today's talk is to decode what machine learning is and how to use it in biological/biomedical research. So first, I'll define some terms, go over some methods in machine learning, then techniques, followed by uses. We'll also see some software tools, and of course, worked examples. Please take the pretest before moving on, and if you want to follow along with the R-code examples later on, the link is shown here and then in the description below. So what is machine learning? Well, here are some different definitions. Machine learning is a branch of AI which focuses on the use of data and algorithms to imitate the way that humans learn, gradually improving its accuracy. Machine learning is a process of using mathematical models of data to help a computer learn without direct instruction, and machine learning is a data analysis technique that teaches computers to do what comes naturally to humans and animals, learn from experience. In summary, it's a collection of mathematical models or algorithms, however you want to say it, that runs in such a way to mimic natural learning. Now, the biggest take-home point here is that the word learning here is analogical, as machines can't actually learn. Furthermore, a lot of machine learning involves processes that are by design hidden, black box models, as it were. So it's important to interpret results carefully. For example, machine learning may find useful patterns in large data sets, say that people who buy applesauce and sippy cups tend to also buy diapers. The results won't tell you why there might be a pattern. So it's up to the researcher to piece that together. So in our example, it's probably because those people buying sippy cups and diapers and applesauce are parents of young children. So before moving on to more terms, I also want to discuss two related concepts, deep learning and neural networks. So the first, deep learning, is a subfield of machine learning. It's less dependent on human intervention to learning, and it's based on neural networks. So neural networks, in turn, are a subfield composing of layers, hidden and not, that are essentially a collection of loosely connected nodes that represent neuron connectivity. So again, it's loosely representing the vast complexity and sophistication of biological brains. Brains are not reducible to computers. Beep, bop, boop. On to terms. Let me translate some common terms used in machine learning to their standard statistical nomenclature. So we have labels, which is the dependent variable in statistics. And then a feature will be the independent variables. So if you have your equation like this, y, and you're trying to predict y with a series of x's, so this will be the label, where these will be the features. Terms like feature creation is another way to say transformation in normal statistics. And then classes are mutually exclusive groups, whereas labels are not mutually exclusive. Okay, with data sets, unlike in standard statistics where you typically analyze the whole data set, machine learning typically splits the data into 2 or 3 subsets. So there is the training set, which is essentially used to train the model. So that's a subset there. It's usually the bulk of it. Then there is the validation set. Make sure the model is not overfitted, etc. That will be a smaller portion of it. And then the testing set used for actual research questions, determining the accuracy of the model. Now, of the 3, the validation data set isn't always used. Sometimes it's just training and test. And there's other ways to sort of partition out than just splitting like this. Sometimes there's like cross-validation, etc. Okay, whirl, on to methods. There are 2 major methods. I would point supervised and unsupervised, and then 2 sort of variant methods, semi-supervised and reinforcement. So between supervised, and this is kind of a schematic of supervised, unsupervised, and reinforcement. And between unsupervised and supervised, there are 3 major uses, clustering, classification, and regression. So with supervised, it's the use of labeled datasets to train algorithms to classify data or predict outcomes accurately. So you see that supervised here, you have input, you're going to have some sort of training and then some sort of output. Unsupervised, it's use of algorithms to analyze and cluster unlabeled datasets. So you see it's right here, input, there's no sort of training, it's just sort of unsupervised, and then you bloop out some output here. Again, this is another way to show it. So you have unsupervised and supervised. Again, you can see that unsupervised goes into clustering. Also, we'll see dimensional reduction. And then supervised learning is like classification and regression that can be used for. So again, the main difference between the two is the use of labeled data. So supervised have data with labels and input have data without labels. And semi-supervised is where it comes in. It's a happy medium between the two. During training, a smaller labeled data set guides classification and feature extraction from a larger unlabeled data set. And then reinforcement learning is sort of like the supervised. We can kind of see over here, we see this sort of feedback loop. It releases feedback, but it's not necessary for each input or state. And then the ideal algorithm can learn how to make decisions in an uncertain environment. So there's going to be some sort of reinforcement signal to say, yeah, that was that did a good job, or it wasn't particularly accurate. So I'm going to be focusing more on just supervised and unsupervised learning as we move forward, because kind of reinforcement learning is sort of like a subset of supervised, and then semi-supervised is sort of a mix between these two. Anyways, so to actually run machine learning, the basic overview is something like this. You're going to have to collect and prepare your data. A lot of times this data is large, so you're going to need to clean it up, format it, whatever. You're going to need to split it up and then like train the model. And you're going to validate the model, make sure it's accurate, and then interpret whatever your results coming out. So other schemas will have slightly different features. Here's one example where they actually have 5 steps, or go into more detail. This deals a lot with more processing and algorithms and sort of a lot of this. You can see a lot of this. These sort of steps are going to be cyclical. There's going to be a lot of steps involved, going through it, going back and forth, etc. But the basic idea is going to be the same. You're going to have data, you train, test, interpret. Okay, on actual techniques used, again, I'm going to focus on supervised and unsupervised methods, as semi-supervised and reinforcement kind of bleeds into those other methods. So in supervised learning, there are all sorts of things we can do here: neural networks, genetic algorithms, native Bayesian networks, linear regression, logistic regression, random forests, support vector machines, decision trees, gradient boosting and bagging, multivariate adaptation regression, splines, and nearest neighbor. Unsupervised, there's also things like neural networks and genetic algorithms. So you can see here, the same techniques can be used for different types of learning, so depending on specifically what you want out. So this unsupervised can also use Bayesian networks, PCA, that's principal components analysis, so that's dimensional reduction, and then singular value decomposition, a lot of clustering, mixture models, self-organizing maps, association sequence discovery, expectation maximization, Bayesian networks again, so that was a replication, kernel density estimation, sequential covering, rule building. So again, here's kind of the big picture stuff. Supervised learning can be classification, regression. Unsupervised learning can be clustering, and it's not really well shown here, but you can also have, dimensional. I'm just going to go dimensional reduction. So DR. Those sort of techniques like PCA. All right. We've got out of methods and techniques. What can we actually use them for? Applications. So conceptually, you can do things like predict values, identify unusual occurrences, and find structure and predict categories. Now remember, computers can't actually learn, so you'll need to provide the brains to the operation. Don't just throw a bunch of stuff into a technique and sift through it. Now, for general, so read here, non-life science applications. Here are some kind of buzzy topics that you might hear in conjunction with machine learning, like speech recognition, customer service, computer vision, recommendation engines, and automatic stock trading. And so, like I said, these are kind of buzzwordy, but they are where a lot of the surprisingly hard problems do lurk. But on the more healthcare biomedical side, we have things like diagnostic tools, patient monitoring and outbreak predictions, tumor detection, drug discovery, DNA sequencing, define coding regions, structure prediction, neural networks to classify cellular images, genome analysis, drug discovery, and then even AI in healthcare. And you can even do drug manufacturing, personalized medicine, and stroke diagnosis. So these are just some of the things you can do. Now, for biology as a whole, we can think in terms of bioinformatics and computational biology. So bioinformatics seeks to solve biological issues by assessing and analyzing and interpreting biodata, whereas computational biology is concerned with finding solutions to issues that arise from bioinformatics studies. So with next generational sequencing, we now have fire hoses of omics data that machine learning can be marshaled to fight. So here are some examples. So in genomics, we can have regulatory genomics and all its things, structural genomics, functional genomics, genome sequencing, gene editing in clinical workflow. I've provided in parentheses more specific examples. Proteomics, you have things like mass spectral peaks, protein recognition by sequence database searching, etc. Microarrays, be able to spot significant interactions in all those complex environments, classifying genes, clustering, gene analysis, differentiating gene states, projecting future gene changes, and the works. And then systems biology, capturing interactions between different biological components at different systems and looking at the whole system's behavior. That has a whole host of things you can do: graphical modeling, genetic algorithms, and Markov chain optimization. So all those applications are great, but how do you actually do machine learning? Well, with machines, aka software. So there's certainly software programs for specific applications like Cell Profiler, Deep Variant, Atomwise, TensorFlow, or you can work in your favorite statistical software language. So SAS, if you want to do that, it can get real fancy with machine learning. Here's a cheat sheet, for example. But you can also do a bunch in base SAS. So here is a whole host of procedures that can deal with machine learning. Now, if SAS is not your style, Python is another heavy machine learning hitter with libraries such as NumPy, SciPy, scikit-learn, Theano, TensorFlow, Keras, PyTorch, Pandas, and Matplotlib. Julia, the hip new language on the block, also has a lot of libraries like MLJ, scikit-learn. You can see how it has a lot of analogs, so scikit-learn, GLM, Decision Tree, Mocha, Net, Flux, Merlin, MLBase, Strata, TensorFlow. And can't be remiss to talk about R, the Swiss Army knife of statistics. It has plenty of packages for setting up data for machine learning, running machine learning, and interpreting and presenting the results. Here are just some examples. Now, I have bolded Caret. That's a particularly good example. That is classification and regression training. It is a mainstay in basic machine learning in R. Those tools not good enough for you? Well, here are even more resources if you want to dig in deep. There's an online course on machine learning. There's a machine learning post in Python. There's actually, this is just the first one, but there's 4 parts. There's a whole book you can find online in machine learning in R. And there's even a book list for machine learning in R. And if you want to get to SAS, presentation slides in machine learning. Okay, time for some machine learning in action. I'll go over 3 simple examples in R. They'll be on classification, regression, and clustering. We'll do classifying tumors, regression decision trees for tumors. These are both supervised, and then k-means clustering for tumors, which is unsupervised. We'll use the same data set, biopsy. So this is biopsy data on breast cancer patients. It has a bunch of these variables here, 8 and 9. Ranges from 1 to 10, and then its class, benign or malignant. So it'd be really helpful and useful if we could use all these variables that we measure to classify a biopsy as benign or malignant. So we'll look at doing just that. So in example 1.1, let's get started. We'll get in and check data. So our data is data biopsy. We'll get ahead of that. Apply this. We want to check what type of data we have. And then we can see here, these are all the variables. These are all integers. I think for our first part here, we want to treat these as numeric. So we're going to exclude some missing data because there's that, and then set variables to numeric. So we're going to exclude that. We're actually going to cut out character because we won't need that. Apply it, make it numeric, and check it again. And okay, this all looks good, numeric, and then our class is our factor. So let's validate our data. We're going to create a validation index, and essentially select 20 for validation, and then the rest for training and testing the models. And there we go. Next here, we'll run algorithms using tenfold cross-validation. So I have a control and a metric, and then that will be plugged in to 5 different classification models that we can test. We're going to set seeds on each of them so they're replicable if you want to use it. So we'll do an LDA, RPart, KNN, SVM radial, and then RF, random forest. Run all of those. From there, we can summarize the accuracy of the models. We're going to save it into a results, all the resampling, and then look at a summary of that. So you can see for all the 5 different models, different accuracies, kappas, etc. But a little more helpful way to look at this is a dot plot of the results. So you can see how accurate they are, how the kappa. And we can see here that in our example, looks like the RF, the random forests, is the best. So we're going to print out that outcome just itself. You can see here. And then we're, let's see here, yeah. So we had the different accuracy, kappa. And then let's make some predictions, given our best model was a random forest. So we'll do predictions, predict using our fitted model, and then our validation data. And then the best way to show this is a confusion matrix. That is a common way to show this. So we found that in predicting whether it's benign or malignant tumors, the accuracy was over 95%. So that's pretty good there. And there's also other characters like sensitivity, specificity, etc. But we'll kind of focus on accuracy. So we had a fairly highly accurate model given the information we plugged in to correctly classify a benign or malignant tumor, which is great. That's what we were hoping to do. Right, let's try a second example. Now doing a regression decision tree for these tumors. So again, check the data, really going to be the same as what we did before. We're going to split into training and testing rather than the training, testing, and validation. So I'm just going to do this sample setup and then probability. So I'm going to have 75% in the training and 25% in the testing. So again, train and test. That's all we need to do here. Now let us model. We're going to do this decision tree using the RPart function. So class and then all those 9 variables using the train data method class. And then we can do a plot. And so this, because it gives our decision tree, this is not the best tree, sort of a default. What we can also use is this function, fancy RPart plot from a package, which is a little bit nicer. And we show here a little bit cleaner setup. First thing to note, looks like variable 2, 1, and 3 were the most useful in diagnosing between benign and malignant. The percentages or the decimals in the second row kind of show the split between benign and malignant. And whatever one was the more prominent one has the label up top. So starting our data set had about 2/3 benign. And then for those that had variable 2 less than 2.5, almost all of them were benign, but a few of them were malignant. And then it was further distinguished by variable 1. And we get down here, only Very few were malignant of that, but all of the ones that had were not smaller than 6.5 were almost all were malignant, and almost all were benign if they had that characteristic. Same thing over here with malignant. Most of them on this side were malignant, so variable 2 really distinguished them out pretty quickly. But then looking at variable 3, if it was less than 2.5, most were benign. So you can see how that splits out, and then all these percentage points should come out equal. So the 100% here, 50, and again, of the 60%, 59% were benign here, and 1% were malignant. And malignant here, or at least initially of these ones, 36 ended up being malignant and 3% benign. So these decision trees take a little bit of interpretation, but they're pretty useful outcomes. Now, you can also look at feature importance. You can use this var imp and see how important features were. Turns out, features 2, 3 were very important. Looks like 5 and 6 were. 5, 6, and 7 were fairly important, but they didn't pitch out here, so they maybe were overlapping. 2 and 3, though, was interesting, where variable 1 didn't have a lot of importance, but it still helped split out this subset. Anyways, you can always dig more into that sort of stuff. But in addition to decision trees, what we really would like out of this, like we did for the other example 1, is to make predictions. So we can make predictions, again fitted against the test, and then do headed predictions, and then again a confusion matrix. We can see here this wasn't as accurate, 92%. It was still pretty decent, but that was another way of getting predictions out. Okay, we're going to turn to our unsupervised learning setup and to do k-means clustering for these tumors. So we're going to get the data in, the biopsy, set it as numeric, and then now, because we don't actually want those class pairs of benign and malignant, I'm going to keep that out of our data set, and then we're just going to cluster using k-means. And because we know we just have 2 categories, the benign or malignant, I'm going to set the cluster number to 2. Obviously, if you had no idea how many classes you're looking for, you can look at different setups of that. I'm going to run that, and then we can graph. So here we go. We end up getting 2 clusters here, which we are expecting that one of these clusters is going to be benign, the other malignant. Looks like they separate fairly well, though, down this area, this sort of space. There is definitely some overlap. And then you can actually look at the optimal clusters, and a silhouette method, I think, is the nicest. And we can see here that, indeed, 2 clusters looks to be the optimal number, which is good because we only have 2 clusters we think are there. So that kind of reinforces that this is accurately telling us that we're trying to distinguish between malignant and benign. Well, that was a fairly rapid and hopefully not terribly painful introduction to machine learning. Obviously, I breezed over a lot of the more complex details that I don't really understand underneath the hood, and the worked examples were fairly simplistic, but hopefully gives you a bit of the working knowledge of what goes into these things, and much more complicated models that you need to build up and test and train, etc. But what we learned here today, hopefully, is that machine learning has many applications in biological and biomedical research. There are lots of techniques. I just looked at very simple ones, but they can all be run using popular software. I showed you R, but SAS, Julia, Python, the software world is your oyster. And actually, turns out it might not be as hard as you might think. Now, ideally, machine learning is for things with predictive applications, whereas some things like inferential applications, really your standard statistical methods are probably good for that. Now, it'd be helpful if you would take the post-test in the survey to see how well this training session covered the material and improved your knowledge. And speaking of improving knowledge, here is a whole slew of references of people who know machine learning much better than I do. It's just part 1. Here is part 2 in the reference, and they're available in the description below. Please acknowledge the DaCCoTA if you find this module useful. Thank you for joining me, and may no spanners be thrown into your works.